In this video, we're going to look at some of the metabolic fates of pyruvate. So if we recall, the pathway glycolysis converts one molecule of glucose into two molecules of pyruvate. So what happens to the pyruvate once it is generated? Well, in animal cells and in us as well, we generally have oxygen present. So the pyruvate, when oxygen is present, is converted ultimately into carbon dioxide and water. And this process occurs through a couple of different pathways. One is the citric acid cycle, and then another is the electron transport chain. And we'll talk about those in detail later. But this conversion of pyruvate to carbon dioxide and water generates a lot of ATP, so it's a very effective way of generating energy. However, in some organisms, such as yeast, the pyruvate is converted into ethanol. Now, why is it converted to ethanol? There's a very important reason for that, and that's what we want to talk a little bit about. Also, animal cells can exist or can undergo conditions where oxygen levels are very low. For example, if someone is doing very strenuous exercise, maybe running a marathon or something, oxygen levels within the tissue can drop. Under those conditions, what are called anaerobic conditions, where glycolysis is occurring in the presence, or rather in the absence of oxygen, pyruvate can be converted to lactate. Now, why is it converted to lactate? Again, there's a very important reason. And just another note, if you've ever done strenuous exercise, or maybe you've watched someone running a marathon or a triathlon, a lot of times their legs can get very wobbly. There's a burning sensation in the muscles. A lot of that is because their oxygen levels are dropping within the tissues, and their pyruvate is being converted to lactate. The lactate is then converted to lactic acid. Lactic acid builds up in the muscles, and it can be toxic, and also can cause some, some sensations of tingling and such as that. So the question we want to answer here: why is pyruvate converted into ethanol in some organisms, and why is it converted to lactate in other organisms under conditions of low oxygen, anaerobic conditions? Okay. So if we look up a little bit closer at the two pathways we were discussing, pyruvate converted to lactate and pyruvate converted to ethanol. Pyruvate is first converted into a molecule called acetaldehyde and then to ethanol. If you notice both of these pathways, again, depending on which organism we're talking about, both of these take NADH. Talked about that previously, nicotinamide adenine dinucleotide. This is the reduced form, and they pull the electrons off, so they're oxidizing it back to NAD+. So they're regenerating NAD+. So this occurs in animal cells under low oxygen conditions, and in things like yeast under anaerobic conditions. The conversion to ethanol, again, it takes the NADH and it oxidizes it, so it regenerates the NAD+. And that's the key here. These two pathways occur, again, in different organisms, but the reason these pathways occur is to regenerate NAD+. It's very critical for glycolysis that we have NAD+. So let's mention a little bit about that. So glycolysis, for glycolysis to occur, there's an absolute requirement for NAD+. If we look back at the steps of glycolysis, we have the first 5 steps, and if we look at from step 5 to 6, if we go back and review glycolysis from 5 to 6, NAD+ was converted to NADH, right? So we were reducing NAD+ to form NADH. Now there's a limited supply of NAD+ within our cells. So without oxygen present, eventually all of the NAD+ is converted to NADH, and glycolysis will stop occurring. We're not going to be able to produce any more energy, and that could be lethal. So the pathways that we were discussing, the conversion of pyruvate to lactate, for example, the reason that step occurs is because there's not enough oxygen to undergo oxidative processes, and so the NAD+ must be regenerated through the production of lactate. So we said that glycolysis has an absolute requirement for NAD+. During anaerobic operations, such as fermentation or anaerobic glycolysis, NAD+ must be regenerated in the cytosol, or glycolysis will slow or stop. Now, under aerobic conditions, when oxygen is present, this is not a big problem because, as we said, pyruvate ultimately goes through the citric acid cycle, through the electron transport chain, and the electron transport chain, which we'll talk about again a little later, is very effective at regenerating that NAD+. The NADH, those electrons are pulled off, sent down the electron transport chain, so we can regenerate NAD+, no problem when oxygen's present. But when oxygen's not present, we have to convert that pyruvate to lactate in order to get the NAD+ to be regenerated. So here's that reaction in just a little more detail. Here we have the pyruvate, NADH, and that's converted to lactate, NAD+. This is a very exergonic reaction. And this reaction is catalyzed by an enzyme called lactate dehydrogenase. Now, just a little bit about lactate dehydrogenase. It's an interesting enzyme. It contains different isoforms or isozymes. Isoforms and isozymes, they're basically an enzyme, different forms of an enzyme that catalyze the same reaction. Lactate dehydrogenase is known to have 5 different isozymes or isoforms, and they're made up of combinations of these two different subunits. We have either an H subunit or an M subunit, and you have the different isozymes made up of different combinations, as you can see here. So these isozymes all catalyze the same chemical reaction, but they have different sequences. LDH, lactate dehydrogenase, is a tetrameric, meaning 4 subunits, protein consisting of M and H subunit with different slight differences in their sequence. So we also looked at an isoform or isozyme with hexokinase. There were some different forms of that. So these different isozymes have different KMs, and so therefore they can function at different levels of lactate. So that's just a little bit about some of the different fates of pyruvate. What we're going to focus mostly on is the direction of lactate when oxygen is present, so aerobic respiration, where we'll go through the citric acid cycle and ultimately the electron transport chain.